A very good afternoon to one and all. A warm welcome to the Honorable Resource Person, Professor Mukesh Doble, Worthy Principal Sir, Dr. Krishan Kant Gupta, HOD cum Coordinator, Dr. Poonam Anand, my fellow colleagues, all the participants from various colleges, universities, and various research institutions across the nation. Before we start further, we wish to seek blessings of Ma Saraswati, Goddess of Wisdom and Learning, with Saraswati Vandana.
श्वेतपद्मासना या ब्रह्माच्युतशङ्करप्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा शुक्लां ब्रह्मविचारसारपरमा आद्यां जगद्व्यापिनीं वीणापुस्तकधारिणीम् अभयदां जाड्यान्धकारापहां हस्ते स्फाटिकमालिकां विदधतीं पद्मासने संस्थितां वन्दे तां परमेश्वरीं भगवतीं बुद्धिप्रदां शारदाम्।
I, Dr. Mino Agrawal, convener of this workshop, welcome you all for this National Lab Workshop on Computer-Aided Drug Design. At the outset, I would like to convey my sincere thanks to our respected Principal Sir for providing us opportunity to organize this workshop. His hard work attitude has always motivated us to keep our spirits high. We feel fortunate to have him at this inaugural session of this workshop. We welcome you, sir. Drug discovery and development is a time-consuming and expensive process, taking about almost 10 years to come into the market and costing about US $1 billion. Several drugs that enter clinical trials, they fail because of several reasons. In this workshop, we will learn how to design the drugs with the help of computers. So computers will be an aiding tool in drug design. This workshop will also cover drug likeness property like ADME properties, QSAR properties, and most importantly, molecular docking using several softwares that are freely available. They are all web server based, so you upload your problem and the results will come into your email ID and you can start analyzing them. So all they are free of charge, and then all these softwares are extremely good. So we can use it for research purposes. We do not need to go for any commercial software. For this workshop, we have with us Emeritus Professor Mukesh Doble, Department of Biotechnology, IIT Madras, and also Fellow of Royal Society of Chemistry, London, and Director of 2 startup companies in biotechnology. His areas of interest are biomaterials, drug design, bioreactors, and bioprocess. He has authored 320 technical papers, 10 books, filed 15 patents. Out of them are 2 US patents with an H index of 56 and I-10 index of 203. When I approached him and requested to conduct this workshop on CAD, he kindly accepted our invitation, showing his passion for research. Thank you so much, sir. With these words, I would like to invite HOD cum coordinator, Dr. Poonam Anand, to deliver the formal welcome address. Over to you, ma'am.
Thank you, Minu. A very good afternoon to one and all. First of all, a floral welcome to all. Kindly accept our floral welcome. Thank you. Respected Professor Mukesh Doble, resource person of this 4-day workshop, Honorable Principal Sir, Dr. Krishnakant ji, worthy delegates from Pan India, I wish to extend a very warm welcome to you all on behalf of Department of Chemistry, Agrawal College, Vallabhgarh. Let me introduce our resource person to the gathering. Professor Mukesh Doble holds BTech and MTech degree in Chemical Engineering from IIT Madras and PhD degree from the University of Eastern, United Kingdom. He has postdoctoral experience in Texas, USA, and he has worked for 25 years in Imperial Chemical Industries, General Electric Technical Centers, and KCP Limited. And for the last 20 years, he is Emeritus Professor in Department of Biotechnology at Madras. He is recipient of Hardinian Award for Excellence in Basic Research and National Award for Technical Innovation in field of petrochemicals. Would you please mute yourself? He won a national award for technical innovation in field of petrochemicals and downstream plastic processing industry, Government of India, for the innovation of antimicrobial food wrap. Sir, we are grateful to you for accepting our invite. Thank you, sir. Now I express my gratitude to our principal for sparing his valuable time from his busy schedule. Dr. Krishnakant ji is a renowned physicist with over 300 research papers. To his credit, he has chaired a large number of conferences and seminars as keynote speakers, and he is associated with many prestigious national and international bodies. He is recipient of many reputed awards. Besides an academician, he is an excellent administrator too. Sir, I would like to share that ours is a college. This is a postgraduate institution affiliated to MD University Rohtak, and it has strength of 5,000 plus students. Our college has been accredited A++ grade with CGPA score of 3.57. UGC has awarded the college with CP status and Paramarsh scheme for mentoring colleges going for NAC assessment and accreditation. It is his vision and able leadership we could achieve all this. And one more feather in our cap I would like to share. We are proud to share this news that recently one of our students, Manish Narwal, he won gold medal in 15-meter pistol tournament in Tokyo Paralympics 2020. Sir, I would like to express thanks for being with us today and at every step for your constant support and motivation in our all endeavors. Now, a warm welcome to delegates who have shown keen interest in joining us for this workshop. It is going to be a real value addition to us all. Thanks to Dr. Mino Agrawal, convener of this workshop, and organizing committee for working tirelessly for the success of this workshop. Thank you, one and all. Over to Dr. Mino Agrawal.
Thank you, ma'am. Let me take this privilege to introduce our honorable principal, sir, also patron of this workshop, Dr. Krishnakant Gupta. Sir is a renowned physicist and has been the patron of Nuclear Track Society of India from 2015 to 17 and presently a member of National Assessment and Accreditation Council. He has to his credit more than 300 publications, 9 books, 2 patents, and 5 research projects from UGC, BART Mumbai, and DST. He has been a recipient of academic awards and honors, and recently he has been honored with the Bharat Shiksha Award. He has been also honored with the Best Principal Award for 2 times. Words are not enough to describe his achievements. Our college is scaling new heights under the leadership of our visionary principals. Now, I would like to request our respected principal, sir, to shower his blessings by his inaugural address. Over to you, sir.
Thank you, Minu. Am I audible?
Yes, sir, you're audible.
At the outset, I would congratulate the Department of Chemistry in our college, headed by Dr. Poonam Anandji, who is coordinator of this 4 days workshop. The convener, Dr. Mino Agrawal ji, and other departmental colleagues who could dream this initiative. They chased it and they realized it today. And we are here on this platform. And this is the inaugural session of this 4 days workshop. And the speaker, Professor Mukesh Dobleji, who is a very senior scientist and very senior professor, Department of Biotechnology, IIT Madras. I was just listening to Dr. Mino Agrawal, who described his profile, several hundred publications, many patents, many books, and a great contribution in this related field. The workshop on computer-aided drug design, it seems to be highly specific, but simultaneously very useful, particularly for chemists who are there on board with us today from various universities and colleges. And more than 40 participants have already joined this webinar. Maybe it is the attraction of the organizers or the attraction of the speaker or the both. But the number is quite large, because ideal strength for workshop, and that too, as Minu was telling me, that sir will be giving practical demonstrations and everybody needs a laptop. So managing 40 or more than 40 teachers at a time, it is not an easy task. But I know that since we have a highly skilled person as a speaker, so everything will go smoothly, and there will be many takeaways for the chemists, for the associate professors, professors and assistant professors who are attending this workshop. And as I have been told that this workshop is spread over 4 days, truly this is a need of the time, because it is touch me not environment, as I always say. We can see each other, but we cannot touch. Even to touch the laptops and other gadgets, the people are quite afraid of, because of the invisible enemy in the name of coronavirus. But definitely, computer-aided drug design. What to talk of drug design? I think computer is an aiding agent in all walks of life. We would not have survived, or I can say the education would not have survived in Corona period without the aid of technology, and that too computers. So as regards this drug design, this is a highly specific field, and since I am a student of physics, I don't know chemistry, though I studied it in my BSc and 10+2. But gone are the days. Now only administration, not in touch with the subject. But definitely I can foresee the outcomes of this 4 days workshop. It will be very useful, and it will open
सर आपका म्यूट हो गया लगता है.
I am sure that whatever the participants will learn during this 4 days workshop, they will take the ideas to their labs and departments, and they will discuss with their colleagues, and a new culture of research, maybe in colleges or universities, will start. Of course, we will continuously seeking the help and guidance from our learned speaker in future also. I will request Minu to be in touch with Professor Doble so that his services, his expertise can be utilized in future also.
Yes.
I wish the Department of Chemistry all the best, and once again congratulate for organizing such a wonderful workshop spread over 4 days. And once again, I welcome and thank our learned speaker, Professor Doble, on my own behalf, on behalf of Agrawal College, Ballabgarh, on behalf of our management, and on behalf of all the participants. And I also thank the coordinator and the convener for inviting me to this inaugural session, and welcome the speaker and the participants. Thank you very much. Over to Minu, please. Minu.
Thank you, sir, for your words of wisdom. And now kindly permit us to proceed further.
Yeah, please.
Thank you, sir. Now, without wasting time, we are going to start our workshop. We hope you will be benefited and you can make use of all these databases and softwares for furthering your research or furthering your knowledge. Now, I would like to invite worthy Professor Mukesh Doble to begin with the workshop. Over to you, sir.
Thank you very much. Let me thank the principal, head of the Department of Chemistry for organizing this, and let me thank Dr. Mino Agrawal, who had been in touch with me for quite some time when we planned to start this venture actually. So I'm going to spend the next 4 afternoons on computer-aided drug design. Generally, if we had been doing face-to-face, we can sit across and discuss and solve your problems, because as the principal said, we cannot touch each other or see each other. What we have done is that every afternoon we will have the sessions so that you could practice in the morning whatever has been taught. In addition, after every 1 hour, maybe we'll have about 10-15 minutes so that you can practice as well. I don't want you people to practice during the lectures, because you may miss out something very, very important actually. So after every 1 hour, maybe 10-15 minutes, you can practice whatever has been taught. And in addition, every morning sessions, when you are at home, you could also practice whatever has been taught. Okay, so let me show you the can you all see this?
Yes, sir, it's visible.
Okay, so this is my workshop plan. 4 afternoons, I would say. Today, I will talk about drug properties. There are many databases in the web which either give the properties of drugs or small molecules, or it can even calculate the properties, because many of the properties of many compounds might not be available. So there are databases and servers which can do that. And then we will talk about the ADME rules, that is absorption, distribution, metabolism, excretion rules. Then I will talk about how to get these property calculated. So that will be like day 1. So in day 2, we will look at ligand-based drug design. Ligand is your small molecule. They call it ligand, or a lead molecule or a hit molecule. Okay, so it is still not called a drug unless it's approved by the Food and Drug Administration. Okay, so there are two main approaches in ligand-based design. One is called the pharmacophore-based design. The other one is called the quantitative structure activity relation-based design. So we will talk about those two, actually, how to go about doing it, both these approaches. And maybe you can practice in the morning, whatever you have learned. And on the day fourth, we will look at homology modeling. We will look at docking. We'll also look at target prediction. We will also look at molecular modeling. Okay. And if time permits, we'll also look at something called pharmacokinetics. Pharmacokinetics is if a drug is given to a human or an animal, how the body acts on the drug. That's called pharmacokinetics. So we will look at that if time permits, of course, actually. So this is the entire plan of the workshop. And whatever I'm going to show, they are all free softwares, free downloadable softwares or servers, which is accessible for students and faculty. For some of them, you may have to give a mail from your official ID. Then only they will give you a password or a license key. You can download softwares like PyMol, Marvin's Sketch. Okay, these requires your university ID email. Okay, then there is something called Open Babel which you can download. Okay, these are the things actually, so which you need to download, which will be used in day 2, and then in day 3 there is something called a build QSAR, which can be used for building your QSAR model actually. Okay, and otherwise we will be using some servers which are also free. So you upload your information and the output will come through your email. Like that it works actually. Okay, so whatever I'm going to talk about is all free software, so you could use them for your regular teaching to your undergraduate, postgraduate. You could use them for BTech, BSc project, MSc projects. You could use them for some of your research and so on. So there are no commercial softwares I'm going to talk about actually. That's the beauty of this whole workshop. There is a, I hope you have heard about NPTEL. NPTEL is a free online video courses conducted by all the IITs. There is a course on computer-aided drug design of mine. It's a 40 hours course, which gives this software usage as well as lot of background fundas actually. So we will not have time to go through all the fundamental concepts because it's just a 4 afternoon workshop. So we will be doing how to use the software, more of a practical hands-on rather than looking at the basic concepts. So if you attend that course, it's more like a complete 3-credit course, like actually. So if you have time, maybe you can attend. Generally, it comes in the July semester, July to October, November. There are 8 assignments, and then there is one final exam also. We just finished it actually just now. So if you are interested to go through the entire story of computer-aided drug design, trying to understand the fundamental concepts and all that, maybe next July you can attend that. But this workshop is more of hands-on, how to use various softwares, which can help you to design both ligand-based as well as target-based drug design. That's what it is all about actually. Okay, I hope you can see this slide. So if you look at drug discovery, okay, so it starts from the lab. Okay, and then it gets partially validated for the treatment of a specific disease. That's what drug discovery is all about, actually. Okay, so trying to understand the mechanism of action, that means how it acts, what are the enzymes involved, and what are the protein targets involved, in which it works through, and then trying to identify the target protein or target enzyme, or it could be an ion channel, whatever be the target, and then validate the target. That means you have to know the three-dimensional structure of the target. We have to know the active site of the target. All those points we need to know. Then looking at the small molecule, that's the lead. Okay, that's the ligand. Different names are there, lead molecule or ligand, we say, or we call it hit. It's not called a drug. It's called a drug only when it is completely approved by the FDA. Okay, until then, generally we call it a lead molecule, lead identification and optimization. You have to modify the structure so that it not only satisfies the properties as a drug, but also it has to satisfy the properties of the drug likeness property, because ultimately we are taking this drug orally. It goes inside the body and acts, so it should have certain properties which is called drug likeness. We will look at those properties. There are many properties. Okay, so the optimization comes there, actually. And then the ADME properties of the molecule: absorption, distribution, metabolism, excretion. Absorption, distribution, metabolism, excretion. These are the called ADME properties. Because it's if you take an oral drug, it gets absorbed, it goes through the bloodstream, and it also gets metabolized, biotransformed, and finally, after it's done its job, it gets excreted. So these are very important. All the properties has to be very optimal, so that it doesn't harm the body. Okay, that's very important. That's called ADME properties. And then pharmacokinetics, how the body acts on the drug. Okay, how the body acts on the drug. You have liver, which is a xenobiotic gatekeeper, so it keeps degrading whatever comes there. So your drug should not get degraded and get excreted before it is doing its job. So that's pharmacokinetics. Then you have the pharmacodynamics, how the drug acts on the body. And then finally, toxicity. We can't take anything orally because it can have toxicities. Short-term toxicity, long-term toxicity, genotoxicity, nephrotoxicity, skin irritations. So many types of toxicities are possible. So a very good compound might not be used as a drug because of toxicity. Even recently, the past 1 year, you have been talking about, hearing about some antiviral drug. It's having good COVID activity, but it's toxic. So you must have been hearing quite a lot about this toxicity in the past 1 year. So not all compounds can be taken orally as a drug because of this toxicity, actually. Okay. So drug discovery includes all these aspects, actually. So drug discovery does not include clinical studies, preclinical studies, that is animal studies, human studies, approval by regulatory, sales, marketing. These are all called drug development. These are not called drug discovery. Only this top portion is called the drug discovery. So here I'm going to teach you how to use computer-based approach to help you in many of these aspects, actually. Okay, so let's look at some molecules, just for fun. This is called aspirin. We all would have taken aspirin, right? Aspirin is a very common, almost maybe 800 years old, I think. It was used for pain, antipyretic. Then later on, now it's being used for cardiovascular, blood as a blood thinner, even for certain types of cancer. So it's an acetyl salicylic acid. Okay. So it's got a lot of properties, informatics properties. Okay, we call it features, drug features, molecular features, and so on. So some of these properties, like it's got a net -1 charge because the COOH can always dissociate. Okay, it's got hydrogen bond acceptors. Nitrogen, oxygen are called hydrogen bond acceptors, as long as they do not carry any charge. Okay, so we have acceptors here. Okay, when this is not dissociated, then you have in charge. So 4 acceptors. Okay, then total polar surface area. It's polar means more of a hydrophilic. Hydrophilic means more of water loving. Hydrophobic is more of lipophilic or more of a lipid loving. Rotatable bonds, it's got 2 rotatable bonds. Look at this. So this portion can rotate like this. Okay, and then again this portion can rotate like this. So 2 rotatable bonds. Okay, and then molecular weight. These are some properties given. Then there is something called log P. Log P is a very important property. It gives you an idea about the hydrophobic, hydrophilic nature, or lipophilic, hydrophilic nature. Okay, both are very important. Hydrophilic because hydrophilic means water soluble, so solubility has to be there. Once the drug goes to the stomach, it has to get absorbed through the gastrointestinal tract. So it has to be, as you know, the GI tract is very lipophilic because it contains a lot of lipids. So it has to have hydrophobic nature. So you see, a drug has to have both hydrophilic and hydrophobic nature. So log P gives you an idea of that actually. Okay. How is this log P calculated? You take octanol. Octanol is considered as lipophilic. Then you take water. So you take a mixture of octanol, water. You put a molecule in that, shake it, and then leave it for separation. See how much goes into octanol layer, how much goes into water layer. So you take a ratio, okay, that is P, then you take a logarithm of that. That's how log P is calculated. So higher number log P means more lipophilic, that means more hydrophobic. Lower log P means more hydrophilic. So you need to have a balance of both. Actually, that's very, very important. Because if it is too hydrophobic, it will pass through the GI, that is the gastrointestinal tract, reach the bloodstream, but it might not be water soluble. That's a problem. Whereas if it is too hydrophilic, it may be good water soluble, but it might not go through the GI. So there has to be a balance. We will look at those later as we go along actually. So aspirin, some of the properties experimentally, and also another important thing you can see, it is binding to 2 different enzymes called prostaglandin synthase 1, prostaglandin synthase 2. There are 2 enzymes in the inflammatory pathway, okay, in the arachidonic acid inflammatory pathway. So these 2 enzymes convert arachidonic acid into prostaglandin H2, okay. So there are 2 enzymes in that. So these are determined experimentally and stored in the information. So we can get a lot of information about drugs with looking at many of the databases. We'll look at them, each one of them. Okay. Another example. Okay, metformin. Metformin. Nowadays, many people have a problem with the higher blood glucose. Okay, so metformin is a very useful drug which many people take actually. So it's a very hydrophilic molecule. You can see a lot of nitrogen groups are there. So it's quite low, almost negative. That means very, very hydrophilic, highly water soluble. So polar surface area is also very high because a lot of nitrogens are there. Okay, it does not have any hydrogen bond acceptors. Okay, it's got donors. Donors means NH type. You can see a lot of NH groups there. Okay. Okay. These are some top pharmaceutical companies which manufacture drugs. You can see on the right hand side, Novartis, Pfizer, Roche, Sanofi, Merck, Johnson and Johnson, GSK, AstraZeneca. Look at the sales. No, it's a big number, very big number, billions of dollars. These are biopharmaceutical companies. They are not as big as pharmaceutical companies, but they focus only on biopharmaceutical. That is products that are made through biological route. That means like a fermentation or biotransformation like that. Of course, these companies also have nowadays some biotransformation routes. And if you look at antibiotics, antibiotics are made only through fermentation. So you can see big, very big business for pharmaceutical companies. Okay, Indian companies, they're not as big, but some of the Indian companies, Ranbaxy, Reddy's, Lab, Torrent, Cadila, Lupin, Sun Pharma, Nicholas, Dabur, and so on. Actually, these are some top-selling drugs. This is 2019 data. This is an anti-inflammatory. The 2019 data. Yeah, this is an anti-inflammatory used for rheumatoid arthritis. It's called Humira, manufactured by AbbVie. 19.6 billion US dollar sales. That's a huge. This is for cancer. Revlimid is for cancer. This is again oncology. Okay. Johnson and Johnson. This is for a bladder cancer. This is for blood clot. Okay. Molecular degeneration in eye. And this is rheumatoid arthritis or autoimmune. Avastatin, colorectal cancer. Okay. Hutchinson's lymphomania. So most of the drugs you can see in the top 10 selling are related to cancer. And then you have some for anti-inflammatory rheumatoid arthritis. This happens because as the average age of the population increases, you have a lot of age-related disease like rheumatoid arthritis. And of course, there are now drugs, quite a lot of drugs are being looked at for central nervous system disease, Parkinson's, and so on. So predominantly, you can see a lot of cancer related, and then joint pain, arthritis. And these ones that are marked in red are biologics. That means they are made through fermentation route. So rest of them are made through chemical route. So if you say, if you have seen similar top-selling drugs about 7-8 years back, you would have seen most of them made through chemical route. But slowly, more drugs are getting manufactured through biological route. That means biotransformation or fermentation, genetically modified strains, large molecules, large proteins as drug candidates, and so on. So the chemistry of synthesis of small molecules is actually, I would say, going down. Maybe biotransformation, that will be for single step, but predominantly you can see a lot of biologics. And as I said, if you have looked 6-7 years back, you would have seen a lot of small molecules, but now you have the top 10, only 4 small molecules, rest all for biological routes. So chemistry is changing. Okay, so drug discovery is a very time-consuming process, a lot of money. Lot of drugs are tested, many patients are tested, finally it gets approved. Okay, you must have been hearing about drugs for the SARS-CoV-2 also, right? Many patients have been tested, this drug doesn't work, that one doesn't work, that doesn't work, and so on. You must have been hearing. Okay, so same story here. Whereas in SARS-CoV, they were testing existing drug. It's called drug repositioning existing drugs. Whereas when I'm talking about a novel drug that is new candidate, you may have to go through a lot of testing and it's a costly business. Whereas repositioning means existing drug which is approved, you are trying for some other disease. Even that doesn't seem to work very efficient. Okay. So many compounds fail because of so many different reasons. Actually. Okay, so if you look at the drug discovery, why a lot of drugs fail? So there are something called clinical trials, clinical trial 1, clinical trial 2, clinical trial 3, and so on, using human volunteers. So you can see 9 out of every 10 drugs fail in clinical testing. Okay, you can see 32% fail, 21% enter phase 1. So in phase 1, some drugs fail in phase 2, some drugs fail in 3. So 9 out of 10. In fact, it is almost 94% of candidates fail. Okay, so a lot of money is lost by the drug companies because of so many reasons. Actually, we look at it. So you have clinical trial phase 1, phase 2, phase 3. So overall success rate is only 3 to 8% actually. So it is like if you test 100 drugs in the human trials, only 3 pass. Even coming for human trials, you may have to test maybe 10,000, 20,000 drugs. So a lot of expenses. Okay, so many examples of statistics of why drugs fail. I mean, many examples actually. Toxicities reason. Okay. So many reasons are there. Okay, so failure of compounds. 39% fail because of poor biopharmaceutical property. Biopharmaceutical means maybe it is not getting absorbed in the stomach. Maybe it is not dissolving properly. Okay, maybe it is getting degraded by the enzymes, so on. For absorption, for distribution, maybe it gets metabolized, and so on. Maybe about 29% fail because of efficacy. It works very well in your lab, but when you come to animal or human trials, it doesn't seem to work. Third reason, toxicity. Okay, like I said, short-term toxicity, long-term toxicity, nephrotoxicity, genotoxicity, okay, neurotoxicity, so many different types of toxicities, drug fails. So you can add up all these, almost 94% it comes to because of the failure of compounds actually, okay. Okay, so this is an interesting picture. Here I mentioned about the clinical trials, the human volunteer trials, phase 1, phase 2, phase 3. And before clinical trials, generally they are tested on rats or mouse or guinea pig. It's called preclinical trials. Okay. And before that, if you see, we have a computer-aided drug design comes here. We go into lead identification. That is your small molecule drug, or a lead or hit or ligand. So we use things like QSIR, and then finally, of course, you need to validate in your lab. Okay, before that, we have to identify the target. You use a lot of tools like proteomics, bioinformatics tools, and before that, you are trying to look at the mechanism of the disease. For example, if I'm looking at inflammation, what is the pathway of inflammation? What are the enzymes that are involved in the inflammation? That's looking at disease mechanism. So you use tools like computational biology, systems biology here. Once you decide on a particular target protein which you want to block, that's called target identification. We use tools like proteomics. We try to isolate the protein using two-dimensional gel chromatography, and then we use a LC-MS to identify the amino acid sequence and get the two-dimensional the amino acid sequence of the protein. Then we compare the sequence with the existing protein, and we use all the bioinformatics tool. Then if we can crystallize the protein, we can get the three-dimensional structure of the protein using an X-ray diffractor crystallographic instrument. So we get the 3D structure of the protein. So then we look at the small molecule or lead molecule which will go and bind to the target. That's the drug part of it actually. That's the lead discovery. And so my focus of this workshop is more here actually. Lead discovery, lead identification, lead optimization, and so on. Now, simultaneously, because 95% or 94% of drugs fail because of poor biopharmaceutical properties, we have to also identify or determine the drug likeness property, toxicity properties, ADME properties, absorption, distribution, metabolism, excretion. Okay, so they are, of course, determined experimentally in the lab. We can also use computational tools to predict these properties also simultaneously. Of course, ultimately you have to get all experimentally, but simultaneously. So if you think there are some compounds which looks very promising, don't just jump into clinical trials, preclinical and clinical trials, but try to estimate all the drug likeness property using different softwares. And if the properties don't seem to be good, don't waste your time. Okay, the properties look good, you can consider that because it may pass the animal trials. Okay, so simultaneously, when you are working on new molecules in the lab, you must determine their drug likeness property, absorption, distribution, metabolism, excretion property, toxicity properties using different softwares, and don't waste time on those which don't seem to have good properties. Select only those which seem to have acceptable drug likeness properties. Actually. Okay, so this has become part of drug discovery. Whereas once upon a time, what they did was they identified a few molecules which worked very well in lab. They went to the animal studies and then it did not work. So a lot of money is lost actually. So or they worked in animal, when they took it to human, it did not work because of some poor properties. A lot of money was lost actually. So it is very, very important to simultaneously look at this drug likeness property when you are working on activity part of it actually. That's very, very important. Okay, there are a lot of books. If somebody is interested, we can take a screenshot of this. You want to go further into drug discoveries, computer-aided drug discoveries, molecular modeling, and so on. Actually, many databases. I will show you many more than what is shown here. I can share the Word document with you people on some of the, okay. There are many journals also. Yeah, excuse me. Let's not go too much on this. Okay. We need to understand what's happening to the drug we consume orally. We are talking about only oral drugs. Otherwise, generally, there are different routes for drug administration: intravenous, intraperitoneal, intramuscular, intranasal, intravaginal, intraanal. So there are different routes for drug administration, but generally we like to take oral drugs only. So we are talking about oral drugs. So drugs, we take drug orally, and the pH of the stomach is extremely acidic, pH 2. Okay, so your drug has to be very stable at that pH. That's very important. Okay, and it has to be soluble at that pH also. Correct? If it has to get absorbed and go to the bloodstream. Now, as the drug travels down, the pH will slowly increase. Duodenum, okay, then it goes through the small intestine, then it goes to the colon, large intestine, and so on. The pH keeps increasing from almost 2 right up to 6 or 7. So it's becoming more neutral. So the drug has to be stable at all these pH conditions. Drug has to be soluble at all these pH conditions. And once the drug gets absorbed through the bloodstream, it will start acting. But if the drug doesn't get absorbed, it comes out as excretion, which is again a waste, because if some drug is lost as excretion, that's a waste. A drug that gets absorbed in the bloodstream, you know, the pH of the blood is 7.4. Again, the drug has to be soluble and stable at that pH condition. Now, as you know, there is a circulation of drug and it goes through the liver, where a lot of xenobiotics or toxic chemicals are degraded by the liver enzymes. The liver has a lot of enzymes like oxidoreductase, P-glycoprotein type of enzymes. Okay, there are enzymes like lipases, esterases, amidases. So all these enzymes degrade many xenobiotics, foreign compounds. The idea of them is to degrade or make it more hydrophilic so that they can get excreted through the kidney, through the urine actually. So you don't want your drug also to get captured in that, because then if it gets degraded, then some amount of drug is lost, right? So that again, that is very disadvantageous. So the drug has to escape that, and then it's going through the blood plasma. Sometimes there are plasma proteins which can degrade the drug. Okay, sometimes drug gets absorbed in the tissues. Okay, so finally the drug is reaching the target where it is going to act. For example, if I have a swelling in my stomach, in my foot, for example, I take ibuprofen. I hope many of you know what is ibuprofen. Ibuprofen is an anti-inflammatory drug. Okay, non-selective anti. So it has to be soluble at this pH, stable at this pH. Then it gets absorbed, goes to the bloodstream. It should not get degraded in the liver. Then it will go through the plasma. That is, and then sometimes it gets absorbed in the tissues, and then it reaches my foot, and it starts working and reducing the swelling of the foot. So I may take a 500 milligram ibuprofen. What is reaching at my foot may be very, very small to act actually, because it's going through so many steps. So this ratio is called oral bioavailability. Okay, this ratio is called oral bioavailability. Ratio of what is available versus what is given as a dose actually. So good oral bioavailability means most of the drug is reaching the target. Say, suppose if you look at percentage, 60%, 70% is very, very good. 30% is not very good actually. Of course, there are some drugs with 30% oral bioavailability also. If you look at metformin, it's got 70% bioavailability. That's very good. That means 70% of the drug, metformin, which I take orally, reaches my target site actually. Okay, so you see the journey of the drug undergoing so many different difficulties, and by the time it reaches, it's not able to act because the concentration is so low. That is called a lack of efficacy. Okay, so it may work very well in my lab, but when it is given to an animal or human, so many things happen, so it's not sufficiently enough. The bioavailability could be very low actually, correct? So a drug should have many things, not only activity. We all think only it should be very active, but that was how people were thinking. But the past 10 years, they have realized 94-95% of the drugs fail when it is taken from lab to animals or human, because we just focused on activity, but it should have many things. So it fails because of that. Selectivity, going to a single target. It should get absorbed properly. It should get distributed. It should not get metabolized and become useless. It should be excreted so that it doesn't accumulate and become toxic. It should be soluble at pH 2 because, as I said, stomach, and then 7, your neutral blood. It should have enough bioavailability. It should not have side effects. It should not have toxicity. It should not have, even the metabolite should not be toxic because the drugs will ultimately get degraded inside and form small molecule metabolites. So even that should not be toxic. It should be stable at all these pH condition. Okay. Then there is a barrier which separates the brain with the drug, with the rest of the body. It's a very polar barrier. It allows only nutrients and oxygen to reach the brain. It does not allow other molecules, xenobiotics, viruses, and other things to cross, because the brain homeostasis has to be maintained very well. Otherwise, brain will get disturbed in its function. So this blood-brain barrier prevents anything foreign. Okay, your drug should not cross that blood-brain barrier and cause damage or disturb its homeostasis very much. Actually, that's called a blood-brain barrier penetration. Okay, then called PGB efflux. This is called a P-glycoprotein. There are many glycoproteins inside the body which captures any foreign material and throws it out. Okay, the idea is to protect your body from toxic chemicals, xenobiotics, and other things. Okay, there are many of these efflux pumps, they are called, in pregnant uterus, in stomach, in the brain, in many parts of the body. Now, your drug also should not get captured and thrown out. After all, the efflux pumps may think drug as a toxic chemical and throw it out. So that should not happen. So we need to design so that it does not get thrown out actually as a PGB efflux, P-glycoprotein. Okay, and then of course it should remain stable for a long time, because the patient may take the drug after 3 months from the day of manufacture. Then in which form it is given, all these different forms: oral, intravenous, intrapenetratory, intramuscular, and so on. And then mechanism, how it acts, cost. In India, it plays a very important role. Cost is a very important action. So, and then how easy it is to make. Then animal studies, how easy it is to make animal studies. So you see, so many things are very, very important, not just activity. So don't think I make a small molecule and test it out on some cancer cell lines. It works well. Oh, I got a drug. But definitely not, because of all these properties have to be simultaneously satisfied. Otherwise, it might not pass the animal studies or human studies. And as I said, about 94-95% of the compounds fail because they don't satisfy all these conditions. Okay, so you need to keep that point in mind. Okay, as a chemist, you may have control on changing structural aspects, right? Molecular weight, hydrogen bond donors, hydrogen bond acceptors, log P, the dissociation, acidic dissociation, basic, polar surface area, how many heterocyclic groups, aromatic rings, ketone groups, rotatable bonds, crystalline, non-crystalline, ester bonds. All these things you can change. As a chemist, you are very good in synthesizing, so you will do a good job. So when you change all this, many of these things change. It is not that only one thing change when I change one. Many things can change when I change one. Okay, for example, I put in more oxygen groups like hydrogen bond acceptors. So when I put in more oxygen groups, then you are increasing its polar surface area. It's becoming very hydrophilic. So what happens? Maybe absorption or absorption through the stomach may be very poor. Okay. So if you have ester bonds, you are putting in ester bonds. So water soluble, because you have oxygen groups, maybe water absorption is good. But then it can become, it may get degraded by esterases type of enzymes, or lipases type of enzyme. So stability is affected. So as a chemist, you may change one, but it may affect more than one. You need to keep that point in mind. So I change one to improve something, but you may be reducing something else also in addition to activity. So it's like a balance between many things. We need to look at how to balance many of these things with each other. That's very, very important. Good properties, good activity, many of them acceptable, and so on. Actually. Okay, that's very, very. So it's a balance ultimately. Activity and then properties, all these properties. So a very good molecule you worked in your lab and say that this molecule is very good active, but it may have very bad properties. Maybe it degrades very fast. Maybe metabolism happens. Maybe it's not stable at pH 2. Maybe it's not getting water. It's not getting absorbed through the GI. Something. Maybe it's toxic, and so on. Actually, so ultimately it's a balance. It's a balance. We need to balance between both these aspects actually. Okay. Okay. As I said, we can experimentally know some of the properties which experimentally determined. Normally pharmaceutical companies will determine some of them when they come up with a drug candidate. They will try to experimentally determine all these properties. You can use softwares also to estimate some of the properties. I will show you some of them also. Some of the properties are already there. If it is a drug, as I said, drug, when a company is trying to sell a drug and they try to get approval from FDA, that is Food and Drug Administration Authority, they try to get all the properties of that drug. Whereas if it is still not a drug, you will not know the experimental information about all these, all the properties. We get only theoretical estimated information. But when it becomes a drug, all the details about that will be known. And now, of course, as you know, there are only maybe less than 5,000 drugs. Drugs means that's been approved by the Food and Drug Administration Authority. But there are millions of molecules which are still not a drug. So many of the properties might not be experimentally determined, but you can calculate using some of the software. Okay, let's look at it's a database called Zinc 15. It's a very useful database. Okay, it's called Zinc 15 database. We will be using this quite a lot. It contains structures of millions of molecules. Okay, 750 million molecules. It's got structures of 750 million molecules. Okay, it's molecules, not only drugs but also other molecules. Like I said, there are only about 5,000 or odd drugs, commercial drugs, but ligands or molecules there are many. So this database has lot of structures of lot of molecules, and some of the properties of this is also known. So they have that properties. It tells you if the mechanism of action is known or targets is known, it is given. Okay, otherwise it's not given. And they're also purchasable. That means if you are interested in some molecule, some novel molecule, you want either as a chemist, you may be synthesizing that, or if you do, if you are a biologist, you don't have a chemistry expertise, you can even purchase them. So they are called purchasable, as you can see here. Purchasable compounds. Structures of a huge number, 750 million. So out of that, 230 million you can purchase. So this is a very useful database of structures. So we will use this quite a lot in the next few days. Okay, let's look at some structures, substances. We can do a search. Aspirin. Aspirin, everybody would have taken, very common. Everybody knows about it. So structures of aspirin, you can see aspirin is there as acetaminophen. This is slightly modified. It's in the para position, as you can see here. There's a double bond. And then there is aspirin, eugenol ester. Okay, we'll look at each one of them. Aspirin, this is called acetyl salicylic acid, aspirin. Some information about the molecule is given here. You can see molecular formula, number of rings is got, heavy atoms. That means atoms which are not hydrogen. That's called heavy atoms, heteroatoms. That which is not carbon is atoms. Sp3 fraction of sp3 orbital. It gives you hydrogen bond donors. Donors means OH or NH group. As I said, hydrogen bond donors here. OH is there. It does not have an hydrogen bond. Acceptors is 4. Yeah, acceptors is 4. 1, 2, 3. Okay, 4. It's not called a donor because it's going to have acid dissociation. So it will become O minus and H plus. So it does not seem to have. That's why they call it 0 donor. This is the total polar surface area. It's a 66 angstrom square. We will talk about it later actually. Remember that total polar surface. Rotatable bonds. It's got 2 rotatable bonds.
Excuse me.
Yeah, this is one.
Excuse me, sir.
Yeah.
Some participants are asking that slide is not visible.
You can't see the zinc database?
No, sir. It is the balance between activity versus drug-like property.
Oh, only that? You are not able to see the zinc database, is it?
No, slide is different, sir.
Yes. Can you see the zinc database?
Yes, sir. Now it's visible.
Now only it's coming. Yeah. Okay, this is the zinc database.
Can we have a sir? Can we have a full screen of this?
Yeah, this is the full screen. This is the full screen.
Okay, thank you, sir.
This is the full screen actually. So this is called the zinc database. It's got 715 million purchasable compounds actually. So if somebody is interested in purchasing some compound which they think it's promising, then they can purchase it. So we can search for molecules. So here you have the search command. I can take aspirin. Okay, aspirin search. You can see this now. Aspirin.
Yes, sir. Yes, sir.
Yeah. So you have the acetyl salicylic acid. So some of the properties are given here. Molecular weight, log P. I mentioned, hydrophobic, hydrophilic balance, molecular formula, number of rings, heavy atoms, heteroatoms. Then it gives you net charge. Okay, net charge because this here you have the OH could be dissociating at acidic pH. Hydrogen bond donors, acceptors. It's got 4 acceptors. 1, 2, 3, 4. A total polar surface area. We will talk about this quite a lot later actually. And one important point I want to show here. Yeah. There is something called SDF. So we can download the structure of this aspirin 3-dimensional structure through this SDF. We will be doing that quite a lot tomorrow. So this is the SDF format, downloading the structure. We will use it even if you are going to do docking, if you are going to do pharmacophore modeling, so many things. We will be downloading the SDF format. We'll do that tomorrow and the after. And then it gives a lot of other information from where you can buy this. And because aspirin is well known, so its activity targets also is given. If it's a new molecule, they will not know the targets. But as I said, prostaglandin synthase, cyclooxygenase 1, prostaglandin synthase 2, cyclooxygenase 2. And because it's an old molecule, a lot of studies have been done. So a lot of information is there for aspirin. Okay. Then similar acetaminophen. Okay. So you have this. Okay, this is also used quite a lot in pain. Again, some of the details of this are given. Hydrogen bond donors. It says 2 hydrogen bond donors. So this is an hydrogen bond donor here. Okay, this is an hydrogen bond donor, OH and OH. He is given total polar surface area. It's much lower. It's given rotatable bonds. Okay, it's got only one rotatable bond. Okay, this double bonds cannot be rotated. Only one bond can be rotated, and so on. Again, if you want the 3-dimensional structure, we download the SDF format of this compound. As I said, we will use this SDF format tomorrow when we do pharmacophore modeling, when we do docking and other studies actually. Again, acetaminophen is also a old molecule. It gives you vendors, details, all those things actually. Yeah, metformin. Metformin is used as an anti-diabetic. Yeah, metformin. Okay, this is metformin. You see log P is negative because it's very, very hydrophilic. A lot of nitrogens are there. Okay, it's highly water soluble. Absorption is lower because it's very, very hydrophilic type of molecule. Molecular weight is given. If you look at the total polar surface area is also very high with respect to aspirin. It's almost 92. Okay. So many properties are given. Vendors are given. Again, this is an old molecule. This is a drug. So a lot of information will be also. So this zinc database we are going to use quite a lot. So like I said, 750 million structures available for this particular in this database. Interestingly, we can even do a search. Okay, we can even draw structures in the zinc database. Suppose I want to draw a structure. So we can draw structures. If it is not there, you are a chemist. Most of you, I think, are chemistry. So you may be coming up with new structures, and it might not be available in zinc database, right? So you can draw structures, and you can do and then we can search. For example, I want to do a search whether molecules with this substructure are there in the zinc database. I am interested, say, let me put this one. Okay. So imagine I am interested in molecules which have this particular substructure, whether such molecules are there in zinc database. Okay, because I am a chemist, we know that these substructures are very important. So what we can do is, sorry, here, substructure search we can do. Okay, so I am interested to see whether there are structures which has that particular type of structure. One benzene ring, then there is an oxygen ether type of linkage. Okay, so it's coming up, as you can see, it is giving in this reddish color, that substructure which I have drawn. So there are many molecules with that substructure. Okay, many molecules. So zinc database can be used for such studies. And as you know, heterocycles are very important because they seem to be having a lot of oxazoles type. They seem to have a lot of properties, biological properties. So I am trying to see whether there are substructures with these type of heterocycles. Substructures. Okay, so it's coming up with the nitrogen oxygen heterocycle, and zinc database is coming up with a lot of molecules possible. So that's the beauty of that zinc database. We can, if I know a particular pharmacophore or a particular moiety is very important, and I can draw that moiety, and then I can do a substructure search in the zinc database to see whether zinc database has molecules with that particular substructure. Okay. Oh, sorry. Can somebody tell what type of structures are these? Can somebody tell what type of structures this?
So participants are writing in chat box
Chat box.
that it is a cyclic ester.
No, not a cyclic ester. No. So we can do this type of search, substructure search. So we can do this type of searches. It's trying to identify from the 750 million, what are the molecules which have this particular substructure, which is shown, which I have drawn using the drawing box here. Okay, so you have the drawing box here. Or we can search if you know the molecule here, or we can draw if you are a chemist and you are trying to draw new things. If it is there in the database, it will try to show. Okay. Okay, these are ligands because zinc contains 750 million molecules, which are all of them are not drug. Only there are only 5,000 drugs are there. So it will not have a lot of properties. So if you are looking only at drugs, there are databases only for drugs. Okay. So there is a database. Okay, can you see these two databases? One is called drugsdb.com and the other one is called drugs.com. Okay, these are databases which gives you information about only drugs. That means drug when you call it a drug, it is something which has been approved by the Food and Drug Administration. Okay, zinc not only will contain drugs, but it will contain lot of ligands because it contains 750 million compounds, whereas these drugs contain only information about drugs actually. Okay, for example, let's look at okay, remdesivir in the drugs. As you know, remdesivir is a drug. It's got anti-inflammatory property. It's used. So this will give lot of information about remdesivir. Okay. where you can purchase them, and what are the side effects. All those information you can find out. So it gives you information about prescription information. It will give you information about the news on metformin. It will give you information on side effects, warnings, because they are all drugs. So you'll get quite a lot of information about the drug. So if you are interested to look at only drugs, then try to use drugsdb.com, and then you can use drugs.com. And there is one more database which is called FDA database, that is Food and Drug Administration database, which is US Food and Administration database. Yeah. Okay, so this is the Food and Drug Administration of United States of America, and it gives you information about all the drugs that have been registered with them, whether it is approved. Even if the drug failed, it will give you information about the failed drugs also. It will give information about the past drugs also. Past means which have been approved by the FDA. So these are databases which are very, very useful to give information about existing drugs. Okay, so drugs bank. So it gives you a lot of, this is another database related to drug. So all these drugs related database gives you a lot of information about existing drugs, including pharmacological activity, giving about the side effects, giving about targets, giving about prescriptions, giving about approval status. So many information it will try to give. You can see this drug bank gives you a lot of information about the drug. Yeah. This is aspirin. And then pharmacological activity of the drug, associated conditions, therapies, pharmacodynamics, how the drug will act in the body, mechanism of action, how the drug acts in the with the body, and then what are the target proteins on which the drug is acting on. Like I mentioned, the prostaglandin synthase 1, prostaglandin synthase 2. Okay, so it gives you information about it. How it's getting absorbed, which proteins it binds to, how it gets metabolized inside the body. Route of elimination, how it is getting eliminated from the body, clearance, toxicity, lethal doses, acute toxicity. So this database gives you a lot of information about existing drugs, actually, existing commercial drugs. Actually. So if you are interested to work in a particular disease, try to first look at all these databases, the FDA databases, drug bank databases, drug DB databases, drugs.com databases, and just to see what are those drugs which are now being marketed for that particular disease, what has been reported, what are the structures of these, what are the targets, what are the side effects, what are the pharmacological action of these drugs, and so on actually. Yeah, remdesivir, this bind RNA virus infection, okay, included COVID. Of course, it did not work properly for COVID, but at certain conditions of COVID, it seems to work. So a lot of information on this actually. This remdesivir is quite a large molecule. Many properties, weight of the chemical formula, and then pharmacological is indicated for the treatment of adults, how it acts, pharmacodynamics, how it acts, mechanism of action, how it works. So it works in the RNA virus in COVID-19. Targets. So it gives you a target. There is an enzyme called, there is a enzyme called replicase in SARS-CoV-2. It seems to work by binding to that actually. So this is the protein. This is called replicase. And the protein details are also given here. This is the uniprot number. We'll talk about the uniprot number. That's the two-dimensional sequence of the particular protein. Okay, that's called the uniprot number. So a lot of information will be given for drugs. So I showed you the zinc database, which contains millions of molecules. And then we have the drug bank, drugscom, FDA, which give information only about drugs. These three databases give information only about drugs. That is drugs which are in the market, which have been approved. Whereas zinc database will not be able to give that information. Zinc database has the structures of almost 750 million molecules. Okay. Okay. So far, you were able to understand this. You could practice it after some time. Let's go forward. There are some more points we need to keep in mind when you are designing molecules. One is, I mentioned the pH. pH of the stomach is very, very acidic. And as it starts going down inside the pH stomach, then you have the jejunum, okay? And then you have the colon, okay, ileum and the colon. The pH will keep increasing. So the drug has to be stable at all these conditions actually. Also, the area of each of these region is very different. So the absorption also can change. Stomach is only small, very small area for drugs to get absorbed in stomach is very small. Whereas when you go to GGM, that is your small intestine area, is very large. So drugs can get absorbed in mostly in the small intestine rather than in the stomach, because as you can see, area is very low. Okay, much lower. And also the amount of time the drug remains inside is also very small in stomach when compared to jejunum or even small intestine, large intestine. It remains much longer actually. Okay, so drugs mostly will get absorbed in the small intestine, okay, not in the stomach, because it remains very small time in the stomach, and also the surface area is lower actually. So in human stomach, mostly acidic drugs are absorbed. Okay, mostly. Okay, like salicylic acid, aspirin. So if you know, if we have a headache, we take aspirin immediately, it gets absorbed because it is getting absorbed. So they are all called fast-acting drug. So if you have to design a drug fast-acting, like I get a headache, immediately it should work. So we should design more of acidic drugs actually, so that it will quickly get absorbed. Otherwise, most of the basic drugs will get absorbed in the small intestine, which may take much longer time actually. Okay. Okay, so when you design drug, it should be undissociated in the acidic environment. Your pH of the stomach is very, very acidic. So we don't want the drug to get dissociated in the stomach. Okay, so if you design like that, it will be neutral, so it will get absorbed. But if it is going to get dissociated at this pH, it will be polar, so it will not get absorbed. Whereas when it goes down in pH, it will get absorbed. So when you are designing for fast-acting drug, we have to keep that point in mind. It should not get dissociated at very high pH condition. So, whereas slow-acting drug, it can get dissociated. So when the pH goes up, it will be in the undissociated form, more like a basic drugs. Then it will get absorbed down the line actually. Okay, some of the anti-malarial drugs, they will be dissociated in stomach, but it will not be ionized in the intestine. So they will get absorbed actually. Okay, so this is a very important point if you are going to design drugs, fast acting, slow acting, and so on. The pKa have to be adjusted. This is a very important graph. When a drug is taken orally, drug gets absorbed in the stomach. So the concentration in the bloodstream goes up like that, reaches a maximum. Drug is getting dissociated in the bloodstream. It's getting metabolized and excreted. So the concentration will go down like that actually. This is how the concentration increase and goes down for a oral drug. Okay. This tells you how long the drug remains in the bloodstream. Okay. How long it's going to be effective in the bloodstream. Some drugs may go up fast and come down fast. Some drugs may come down slowly. If it is coming down slowly, that means the concentration in the blood is there for a longer time. If it is coming down fast, then concentration in the blood is coming down fast. So the doctor may tell, take drug every 4 hours, every 3 hours. Or if it is remaining for a longer time, doctor may tell, take drug once in 12 hours. So that is how they decide, depending upon this graph. This is called a dose response curve, sorry, the pharmacokinetic graph actually for how the drug remains in the bloodstream in a human or an animal actually. Okay, there are I mentioned. I mentioned that the zinc database has a lot of structures, but properties are not known. Only if it is a drug, properties will be known. but I want to estimate properties of molecules. So what do I do? There are many databases which can be used to measure properties. For example, we have a very important database. It's called the Swiss ADMA, which can calculate property of molecules. Given the structure, it can calculate properties. So it's called the Swiss ADMA. Yeah. Can you all see this? Can you see this? Can you see this? Can you see this database?
Yes, sir. Yes, sir. Swiss admin. Yes.
Oh, yeah, somebody has to tell me, no, because okay, this is a very useful website to calculate physicochemical properties or descriptors, they are called, of molecules. So I am designing a new molecule. I don't know the properties. This is a very useful database. Only if it is a drug, then the properties are reported in all these drug-related database. But I don't know the properties. So what do I do? This is extremely useful, which can calculate properties of molecules. For example, let's take a molecule called aspirin. And imagine I want to measure properties, calculate properties. How do I copy the structure of aspirin into Swiss ADME? There is a notation called smiles. I'm going to explain the smiles later to you. Okay, smiles. We can copy the smiles notation. I will explain it later. And then I go to the Swiss ADME. And I can enter a list of smiles here. So I can copy paste that smiles notation of aspirin. Understand? I will tell you what is the smiles later, but immediately you can do that. Copy the smiles notation and take it to Swiss ADME, and then I copy smiles here, and then I will run this. This is the run command. Run. So it is running and done, and it is this aspirin, acetyl salicylic acid. So it's giving lot of properties. Okay, the Swiss ADME calculates properties of structures. You can even draw structure if, as a chemist, I am planning to draw synthesize a new molecule. You can draw the structure, and then I can run and measure the property. Or I take a molecule from zinc database, I copy the smiles notation, take it to Swiss ADME and run it, and it is giving lot of properties. Okay, it is giving molecular weight, number of heavy atoms, total polar surface area, molecular refractivity, hydrogen bond donors, hydrogen bond acceptors, number of rotatable bonds, log p value, GI absorption. That is, it's very good. BBB, blood brain barrier penetration. Yes, P glycoprotein. Yes, there are many enzymes called cytochrome P enzymes which are present in the liver, which acts as a biotransformation. Do the enzymes act on the on those molecule? No, no, no. It is giving you solubility of the molecule. It is giving you bioavailability 55%, which is very good. It's giving average. I will explain this also. There are many rules which tells you whether the molecule has a drug likeness. There is this. Okay, I will explain that. So a lot of properties can be calculated using this Swiss ADME. As you can see here, lot of rules, lot of properties. Bioavailability is good, and then cytochrome enzymes degrading this molecule. No, no, no. Blood-brain barrier, yes, it is getting penetrated through the blood-brain. GI absorption is very good. Solubility, so many things are given. And then many rules are there. Yeah, we will talk about these rules. I will talk about those rules. I will talk about the spice notation, these two things. So zinc, Swiss ADME is a very useful software for us to do many things. And you can even draw structures. And as a chemist, you may be wanting to synthesize new structures, right? And then you think before synthesizing, you would like to know whether property is good or bad. Imagine you are a chemist and you are trying to synthesize some new molecule. Blindly, right? So I have decided I want to know whether the properties are good or not. So I've drawn, and then I do this, and I do this. It's writing the smiles notation. I said I will tell you what is smiles notation, and then I can say run, run. Oh. Yeah. So this molecule, so it's giving you properties of that molecule. Total polar surface area is low because it's very hydrophobic molecule. So only one nitrogen is there. Okay, so and then the log P is lipophilic, bioavailability 55%, then BBBS. See, some enzymes are going to biotransform this particular system actually. It's bad news. Moderately soluble because it's hydrophobic, it's become moderately soluble. Okay, so solubility has come down, not as good actually. Okay. Okay, this is another software, which can calculate pharmacokinetics and toxicity. Upload smiles here again. stop. Yeah, so this is another software which gives you a lot of toxicity. And CACO is the cell line which is found in the stomach epithelial cell. So it's a measure of permeability of any drug through that CACO cell line. So it gives you a lot of, it's running. It gives you a lot of information about metabolism, toxicity, skin hepatotoxicity, minotoxicity, oral rat toxicity. This is related to the cardiac beat. So many types of information also this gives. So there are many software. These two are very good software, Swiss ADME and this particular PKC CSM. So it's calculated as you can see here. This is running here. Blood-brain barrier penetration, yes, it's penetrating a little bit. Glycoprotein, no. Skin permeability, no. Cytochrome, yeah, no. Metabolism, then excretion, it's running here. It will take a little bit of time. Given 72% logarithm of ml per minute kg. Then it gives you a lot of toxicity related information. Here. So these two, yeah, it's giving some toxicity related information. No, no, no. Okay, so giving toxicity. So I showed you two important property prediction. Swiss ADME gives you a lot of information. Either you can draw the molecule here or we can copy the smiles notation. I said I will explain what is smiles notation later. And then this is another software. It's called TKCSM. The website is given here. Okay, can give a lot of information about toxicity. This ADME does not give toxicity relation. So we can draw new molecules in the Swiss ADME. If as a chemist you are trying to design new molecules, if the structures are available in Zinc database, I can copy the smiles notation, take it to Swiss ADME, okay, and then measure the property, calculate the property using Swiss ADME. So these two softwares are quite useful to calculate the property. This is the website for this ADMET, and this is the website for Swiss ADME. And the Zinc database, this is the website. Any questions? Anybody has any questions? You can practice, maybe 5 minutes, 10 minutes, you can practice.
Yes, participants,
Yeah.
are you having question at this stage?
Or you can just practice for about 5-10 minutes.
Yes, sir. Yes.
So try to use Open Zinc database, all of you, and then take out some structure, okay, some common medicines, then go to Swiss ADME. Here is the website, and then copy the smiles notation. I will explain what is smiles, then run it and see the properties of that, and then you can look at toxicity using this particular, again, this software. Copy the smiles notation. So these two are very useful for that. There are many softwares there. I will share this Word document later with you. Okay, you can just try it out.
Excuse me, one participant was having some problem. Dr. Kuldeep,
What?
uh, excuse me, Dr. Kuldeep, you were having some problem in links in downloading these softwares?
No, there is no downloading. All the softwares are like a web-based, right? You're not downloading anything.
Yes.
They're all web, correct?
Fine, sir.
Yeah.
So one participant is writing QSAR.
Yes, yeah. I'm talking something else. Properties. We are talking about the properties. So QSAR does not come into the picture. Correct, no?
Uh,
Okay.
Hello.
Yeah.
Yeah. Good evening, sir. Actually, in email there was a software build QSAR.
QSAR. I'm
That
not
is
talking.
not down.
I am not talking QSAR today. I am talking about the
Okay.
properties. Please have a look at it. Number one. Number two, please, whatever softwares I said, they are all downloadable. Please read the front page of the web. That will tell you all the details. So you can download.
Other software were downloadable, but this build QSAR, the link was not working.
Yeah, please read it. No.
Yeah, all participants has faced the same problem.
Yeah, yeah. So I was able to download and using. So
excuse me, sir. Excuse me, sir. Please unmute yourself. Excuse me, sir. Please unmute yourself. Sir, your voice is not coming. Participant, sir, has given the break for practice. You can do your practice and let us know if any difficulties with you. Yes, sir. Yes. Please practice Swiss admin.
Can we start?
Yes, sir. Let me ask. Yes, participants, can we go further?
I will talk about rules because Swiss ADME, there are many items called rules. No.
Yes, sir. Please continue, sir.
Yeah. Okay. I will start on rules because if you look at the Swiss ADME, so what happened was there is something called Lipinski's rule of 5. Okay. So Lipinski and researchers working in Pfizer looked at large number of drugs and they said for a molecule to have good oral bioavailability, the molecular weight of the molecule should be less than 500. Number of hydrogen bond acceptors. Acceptor means nitrogen and oxygen should be less than 10. Hydrogen bond donors should be less than 5. Log P should be less than 5. That's what they said actually. They said that if a molecule has to have a good oral bioavailability, look at the molecule, check the molecular weight. If it is less than 500, and hydrogen bond acceptors, that is nitrogen, oxygen less than 10, donors less than 5, log P less than 5, then chances are it will have good oral bioavailability. Oral. Okay, if some of the one of these rule doesn't satisfy or 2 doesn't satisfy, chances are it will not be a good drug with oral bioavailability. Don't be careful. Don't waste your time. Okay, so when you go for animal studies, it might not have good drug likeness property. That was Lipinski's rule actually. Then came another group. They said GOS et al. Rule. They said log P should be in this range. Lipinski said log P less than 5, but these people said some range. They said molar refractivity. What is this molar refractivity? Volume of the molecule into refractive index should be in this range. Molecular weight in this range, and number of atoms 20 to 70. So that is their rule. Okay, that is called GOS et al. Then came another group, Weber. They said number of rotatable bonds should be less than 10. See, rotatable bonds are very important because ultimately the molecule goes and binds to a target. So if it has rotatable bonds, it will nicely fit into the target active site. But too many rotatable bonds, it will be so flexible, it will not remain inside, it will come out also, that the stability will be lost. So there has to be some balance in rotatable bonds. So they said rotatable bonds should be less than 10, polar surface area less than 140 angstrom, total number of hydrogen bond acceptors and donors should be less than 12. So you see, this is another rule. Okay. Then came another group called Muge et al. Rule. They said if you should have at least 2 of these groups for a molecule to be drug. Okay, 2 of this group. But if you have one of those with the star, that is good enough. Also, okay, that is Muge et al. Rule. So they saw how many of these groups are there, and if they at least 2 to 7, then they can be classified as drugs actually. So that is called Muge. And then polar surface area rule I have been talking about, which I also said polar surface area. What happens is if a molecule is very hydrophobic, polar surface area will be very low. So absorption through the GI will be very high because GI is lipophilic. As the polar surface area increases, the absorption will go down dramatically and then reach 0. So ideally, you should have a balance because if it is lipophilic, it will not be water soluble. Okay, so generally they say polar surface area should be around 100 or less than 100 angstrom square actually. Of course, there is one rule which says polar surface area should be less than 140. 140 means somewhere here actually. But ideally, they say around 100, you will have about 50% absorption will be there. So ideally, you should work in this region actually. Okay, so polar surface area determines the absorption in the stomach. And then the blood-brain barrier, which I mentioned that there is a barrier which separates the brain with the rest of the body. It allows only nutrients and oxygen to flow through. So there is a rule which says if molecule has nitrogen plus oxygen less than 6, polar surface area less than 60, molecular weight less than 450, log D less than 1 is equal to 1 to 3, log B greater than 0, then it will not permit blood-brain barrier penetration. Then came P glycoprotein. I said P glycoprotein or efflux transporters, which is present in body. They throw out any xenobiotics, toxic chemicals, foreign molecules out into the kidney for biotransformation. They are called P glycoprotein efflux transporters. So they said if nitrogen plus oxygen greater than 8, molecular weight greater than 400, pKa greater than 4, then it will be acting by the PGB, P glycoprotein. But if it is nitrogen plus oxygen less than 4, molecular weight less than 400, pKa less than 8, chances are it will not be thrown out by the proteins called P glycoprotein. Then there is another rule called Otria rule. They said molecular weight should be less than 300, log P less than 3, number of hydrogen bond donors less than 3, hydrogen bond acceptors less than 3, flexible bond less than 3, polar surface area less than 60. Okay, then came another rule which said Han et al. Rule. So you see, there are so many rules come up with so many different groups of people to say whether a molecule will have the drug likeness property, will have good bioavailability, can have good oral absorption, and so on actually. So what we do is when we draw the structure and when we use a Swiss ADME, for example, Swiss ADME also calculates these rules. I said I will talk about it later. Okay, so Swiss ADME calculates. Okay, Swiss ADME. Yeah, Swiss ADME calculates and gives you. You can see this. Can you see this? Can you all see this?
Yes, sir.
So you see Lipinski's rule, Gauss-et-al rule, Weber rule, Egan rule, Muge rule, all these rules, and says yes, it satisfies. Muge maybe no. So Swiss ADME calculates that also. So
So
these
but excuse me, for drug likeness properties, we have to go for all these rules. If we go for only Lipinski rule, then
Lipinski was very old actually. People come up with the new rules actually. That happens, right? So even if you know, they said originally blood pressure should be some 130, 80 normal. Then they said 120, 80 is better. Now they say 135 or 85. It keeps changing, right? So same thing with blood glucose. Originally they said some number. Nowadays they say something else. So always based on knowledge, they keep on improving. So Lipinski is very historical, Purana rule actually. But many groups have come up with different rules. So it's good to look at all the rules and try to see how many rules it satisfies. Does it satisfy most of it except one, or does it not satisfy many of the rules? So it's good to look at it. Lipinski is very old rule. Okay. As I talked about, Lipinski said yeah, can you see this? Lipinski's rule said log P should be less than 5, right? Whereas if you look at other rules, they are saying log P should be less than 3. If you look at the Utpira rule, yeah, they say log P less than 3. And then if you look at another rule, so they keep refining based on more data they get. Okay, so log P in this range. So they keep refining, improving based on more drugs coming into. So Lipinski is a very old rule actually. So these rules are coming up with the more stringent condition to improve on it. Okay, so ideally we should look at many of the rules, all these rules, and see does it not satisfy one rule or many more than one rule. That's always good actually. Okay, that's very important, which tells you an idea about the drug likeness property. So don't just stop with Lipinski's alone. Lipinski, as I said, is a very old rule as historically followed actually. So it gives you a bigger range. So try to look at other rules also and try to.
participants are asking that is there any standard rule made by drug authority?
No, there is nothing. There is no rule. If life is so simple, then there will be thousands of drugs. And as I showed you, 96% of molecule which come to animal trials fail actually. So rule is just like that actually. So just because somebody got less than 50% and he failed doesn't mean he'll be a failure in life, right? He might be. There are so many examples in life where there are big success who did not do well in their exams, who failed and so on. So it's exactly like that actually.
We cannot go for one particular rule.
Yeah, ultimately we are looking at absorption. Ultimately we are looking at bioavailability, toxicity, ultimately on human, right? So these are all predictive tools. So when you do a prediction, there is always going to be some fault. So there is nothing like one rule and that is it, right? In some colleges, 50% is called pass. Some colleges, 40% is pass. Is called pass. So that's a different rule. So they follow that rule, right? So some colleges, they give S with the 80% as marks. Some places they give 70%. Some places they give 90%. So it's exactly like that. Rules means they are always broken with a different condition. So there cannot be a single rule, but this gives you an idea about the oral bioavailability. Ultimately, it's the oral bioavailability, ADME, drug likeness property. That's very important. How they are going to act, I cannot take 10,000 molecules and keep doing experimental all those studies. So, but if I have some equation formula, so that at least it will give me some idea about it, so we can use them. That is it. That doesn't mean that it's 100% proof, right? So suppose I have 50 students in my class. I want to select some basketball players. I will say 6 feet tall people may play basketball well, so I will select. That doesn't mean that 6 feet people will play well or 5 feet people will not play well, but at least we need some rules to shortlist candidates. It's like exams. 50% pass is called, 50% mark is called a pass. Why did they select 50%? So it could be anything. So rules are always just an idea for us to make use of. And the idea is, if I have, as a chemist, decided to synthesize some 100 molecule in my lab, I can use these Swiss ADME like softwares and see out of 100, how many look very good drug likeness and ADME, how many seem to be not so good, clearly not so good. There will be clearly good, and there could be in between candidates, right? You will always have both, all three. Clearly not good, don't waste time synthesizing some molecules because it will not have good drug. Clearly good, yes, but in between you can make a choice based on how many rules it satisfies and so on. Okay, so the rules help you as a sort of a screening tool, actually. So there are many rules are there because for drugs, it's not straightforward. It's going into a human system, which has a very complex system with efflux pumps, biotransformations, xenobiotic properties by the liver. All those factors are there. So your drug has to escape all these and finally reach your target. So there is nothing like one single property which will help you. That is why drug discovery is very challenging. It's not 100%, and it's very, very expensive. Okay.
Sir, there is one question that searching for the properties of novel compound is secure on Swiss admin. Participant is asking, is it secure connection? As URL is showing that it is not a secure website.
Yeah. So. You mean somebody else will copy your molecule or something?
Yes, sir. Is asking that if we go for a novel compound, is it safe? Is it secure to search it on Swiss admin? That is asking that it is not a secure website.
Swiss is a very, very, very well approved.
Yes.
There are almost it's been there for a long time. Everybody uses Swiss for sequence comparison, homology modeling. It's one of the best actually. Swiss Institute of Bioinformatics has been there for a very long time. If you are going to think about cracking and trying to get, I will not be able to answer that, but this is one of the best.
It is a very approved website, so you can do work on this website.
It's very approved, and it's a big research center. Swiss Institute of Bioinformatics. They have a lot of software, entire homology modeling, and modeling of target protein. Everybody uses this Swiss. There is no doubt about the quality of this. This is the best. Even commercial softwares will ultimately use this Swiss for homology modeling. Okay, if we have a new molecule and you think it's very, very novel and you have done a patent search, it is not available, then you patent it, no? What you can do, right? You can have a initially invention disclosure, and then you can start spending time on that. So people do that sort of patent actually. Okay, preliminary patent it's called. Then after 1 year, you can file a full patent. So if you think it's totally new, if you are going to check your molecule using the structure to see whether there are similar molecules, even that is not secure then, correct? If I have a new molecule, I have to check whether such molecule is there in has been predicted. So I have to draw the structure of that molecule to do a structure search in various databases. So there itself, if it is not secure, I may be losing that. So you can't be so paranoid. And if you are going to talk to your patent attorney to find out whether such this molecule is very, very novel, then again you are sharing that knowledge with your patent attorney, and he has to do a world patent search and so on for your molecule to see whether it's novel. Then again, that is you are losing. It is going to be not a secure. Okay? Okay, so all these rules are there, which is Lipinski's rule, Gauss et al. Rule, Weber rule, Muget et al. Rule, the total polar surface area, blood-brain barrier, penetration, P glycoprotein effects, then Ukra rule, Han et al. Rule. All these will help you to sort of guess whether the molecule we are designing will have the drug likeness, the ADME properties, toxicity acceptable properties. So if you plan for say 1,000 molecules, you can look at all their properties and say 50 of them are bad with respect to these properties and other 50 look good. Then you can go and start synthesizing those 50. So that's the main advantage. So Swiss ADM is one software I showed you. Then I showed you another one. It is called the PK CSM. This is also very good. It gives you idea about, as you can see, some toxicity also. So there are many softwares that may be available in literature which can help you to calculate. These are all calculated properties because this it's a novel molecule. Nobody would have done property calculations. I mean property experimental determination. Okay, let's go forward. I said I will tell you what is this SMILES. Okay. SMILES is one way by which we can copy. If you remember, we can copy the structural details which I did. You know, you can see this here from Zinc database to other databases. So SMILES is very, very important for copying the structural details, and beauty is it's in a text form. Okay, so you can see it's got capital C, small c, double bond, oxygen, everything actually. So the entire structure can be written in a text form. That's the main advantage of SMILES. Okay, let's look at what is the SMILES all about. SMILES is simplified molecular input line entry system, simplified molecular input line entry system. So atoms are represented by their normal abbreviation. So carbon, nitrogen, oxygen. So we write just capital C, nitrogen, oxygen, phosphorus, sulfur, fluorine, chlorine. Bromine is Br, iodine, like that we write. Chlorine, we write Cl. For other things, we write a square bracket. Gold means Au. Okay, only for this, it is just without any bracket, automatically it's assumed. And then you don't write anything for hydrogen because implicitly it's assumed. So if I write just carbon, it is assumed as CH4. So if I write just O, it is assumed as H2O, water. So for what representing water, I just write O. Understand? So if I want to write methanol, okay, which is CH3OH, I just write CO, capital C, capital O. So simple actually. So if I want to write ethanol, I will write capital C, capital C, capital O. That becomes ethanol. Okay, now how do I write rings? So if I have cyclohexane, I write the carbon C, 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 C 6 times, and then I put 1, 1 and 1, 1 here. That means this carbon is connected to that carbon. Okay, so you start from this carbon, C, 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 then 1. So this carbon and this carbon are connected. So if you look at dioxane, you start from say 1 oxygen, then CCO, then CC, and then you put 1. Here also 1. So this oxygen is connected to this carbon. Very nice, right? So if you have a double bond, then you have to put this equal to sign. Single bond, you don't have to put anything. double bond, you have to put the equal to sign. Triple bond, you put hash sign. And when you put this bracket, then that means there is a branching happens. So for example, if you want to write a propionic acid, 1, 2 carbon, 3 carbon, then there is a double bond O, OH. So you just write CCCO. So this backbone is gone. And then from this C, there is a double bond O. So you just write bracket, double bond O. So that's propionic acid. Now, what do you do for aromatic? Instead of capital C, you write small c. So for aliphatic, you write capital C. For aromatic, you write small c. So benzene, again 6 carbon, one connection, 1, 1, C, 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 but only small c. So small n means aromatic nitrogen. Small O means aromatic oxygen, like that. So pyridine, you start from nitrogen 1, C, 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 C1. Only thing is everything will be in small form. Furan, you start from oxygen, C, 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 1. So this is furan. Correct? So you use small lowercase. Aliphatic, you use uppercase. Okay. You can represent stereochemistry also by putting in slash and backslash. Transition transform. And you can represent a triple bond using hash sign. So C triple bond N. Hash sign. That is what is called miles. So if you go to your molecule aspirin, you can see the. So it is starting from say this carbon. You can see CC, capital C, and there is a branch of double bond O. Okay, then there is an O, capital O, and after that all small, representing this aromatic system. And this C and this C are connected. So these 6 C's are having an aromatic, and then again you have one capital C and O, and this one is double bond O in a branch. You understand? This is what the smiles for aspirin. You don't have to remember because softwares will automatically calculate, but it's good to understand this. Understand? So 2 capital C's represent this carbon and this carbon, and this double bond O in bracket means there is a branch of double bond O, and then this capital O is this chap, and then there are 6 small C's with 1 and 1, so it's a closed aromatic ring, and then you have one capital C and then capital O. Okay, capital C, capital O, and there is a double bond O in a branch here. Okay, this is what is smile. So we copy smiles notation. So you saw that Swiss ADME says give smiles notation. You go to this PKCM, many softwares want this smiles notation. Okay, copy this. So it says, no, provide a smile string. So many softwares will ask for a smile notation. So it's very useful. We go to Zinc database. When you have the structure, it gives you a smile notation. Even if you draw, it automatically calculates. Look at this. So imagine I'm drawing. Look at this. On the top, can you see? It's automatically given the smiles notation for benzene. Can you see that? You can see that?
Yes, sir. That smile is coming. Yes.
Yeah. Yeah. So automatically, you can see so that the carbon is coming. Carbon, smile, capital C came, another capital C came, another capital C came, and then okay, I put an oxygen, so oxygen here, right? From this benzene carbon within a bracket there is a O. H is not there, so it automatically calculates. So we can copy the smiles notation, take it to Swiss ADME if I want to measure the properties of this particular molecule, right? So I copied the smiles notation of this molecule. Yeah, I copied the smiles notation. I went there and then I say run. So smiles notation is a text form. It is very useful because all these softwares ask for smiles notation, so we can copy from that. Okay, so all these properties are given. It also gives you all the rules also. Lipinski's rule, those rules, new way rule, so lot of these rules actually. So we can copy the smiles notation. And then run the ADMET properties of that new molecule which I proved, right? So yeah, it's running. So all these softwares will look for smile notation. So that means smiles notation is very, very useful for you. We can represent a single bond, double bond, triple bond. We can represent branching. We can represent stereochemistry also. Everything represented in a text form. So this is aliphatic. Sorry, this is an aromatic system. So everything can be represented using this smile. So, another interesting thing we could do with the Swiss ADME is imagine I have celecoxib is an anti-inflammatory drug targeting an enzyme called cyclooxygenase 2. It's a selective cyclooxygenase 2 drug. It's in the market. Okay, selective cyclooxygenase 2 drug. Okay, so I want to know the properties. I can copy the smiles notation. Imagine I go to Swiss ADME. I copy the smiles notation. And then I run. Okay, so this is celecoxib. It is a selective cyclooxygenase 2 drug. It's already in the market. Good GI absorption. It's doing solubility is high. Bioavailability is also good. It's satisfying all the rules. Really goes, it's not satisfying like that. Log T is good, 2.6. Cholecystophysiria 86, which is okay. All the rules it is satisfying, and so on. Okay, so now I want to modify the structure of celecoxib, and I want to see whether so this is celecoxib. I want to modify the structure and see whether any properties get changed. Okay. So imagine I'm a chemist. I'm a medicinal chemist, so I think I will do some modification to the structure. And see whether I'm going to change something. Instead of fluorine, can I put chlorine? So medicinal chemistry. Okay, so I can do this, and then I can convert that into smiles and run that new molecule. Okay, and then see whether the how the properties are getting changed. Okay, so I the fluorine, I have made the whole thing as fluorine. Can you see this? Yeah.
Yes, sir.
Yeah. So I made it into chlorine, and then I see whether the properties I am based on my chemistry knowledge and the medicinal chemistry knowledge. Okay, so I made this instead of fluorine, silicoxib is fluorine containing. It's in the commercial market. It's called selective cyclooxygenase 2 drug. It's given for arthritis. So I'm trying to put it into fluorine, and I am trying to see whether all these properties are satisfied. Okay. Interestingly, all the drug likeness rules are good. Log P is there. GA absorption is also high. Okay. Okay. So I can do that sort of studies also using Swiss ADM. So I can take a parent compound, and then I can think of different substituents, changes, and then put the Swiss ADM, put the smiles, convert it into smiles, and then I can do the property calculations. Right? Now I got the smiles of this new compound, which contains all chlorine. You can see all chlorine instead of fluorine, which is the original molecule. And then I can look at the properties and see how it is. We can do that sort of design on the I call it in silico design on the screen to see that. Of course, we are not looking at activity. This softwares don't predict activity, but the properties, it's able to predict properties. So I can use these software, modify the structures, and I can look at the properties which I've been talking about. Hydrogen bond donors, acceptors, polar surface area, log P value, the various rules given here, cytochrome P, metabolism, P glycoprotein, substrate, blood-brain barrier, GI absorption, solubility factors, okay, bioavailability factors, all these lead likeness property, all these could be looked at. Different properties we can calculate for this molecule. Then I can use this software also. This software, I can use this software also, and then look at the toxicity properties of this chlorine substituted compound. Okay, this is the chlorine instead of fluorine substituted compound. I am putting the chlorine substituted compound, and then see how that is there any worries about the toxicities. So I can do that sort of in silico design and measure that. Property predictions. So it's calculating the properties of this new molecule, which has got chlorine, whereas the silicoxib, which is a commercial product, has fluorine. Yeah, it's spanning. Okay, you can see this running. Yeah, so it also gives you the toxicity of the same molecule, no hepatotoxicity, no skin tension, no oral toxicity. Okay, 50 actually. Any doubts so far? We have covered.
Yes, participants, you can ask your queries, if any.
Yeah, we looked at a lot of databases, drugs databases, zinc database, property calculations, Smiles notation, rules, different types of rules.
Yes, participants, any query?
Any doubts you have, you can practice. In fact,
Yes.
I also mentioned that if some one of your relatives, somebody using some drug or medicines, you can just try whether it's there in zinc. You can look at properties using Swiss ADME and the toxicity predictor, just to get a feel actually.
So how many pharmacokinetic properties we have to take? Like if we are working on one particular compound,
Yeah.
and so how many, the minimum, how many we have to take these properties?
No, we are not taking. We are trying to measure what is the property. That's all.
Yes.
It is just giving you the information. What are the properties? So it is giving all the properties like ADME related properties, toxicity related properties. So if we know that it's going to be hepatotoxic, then that's a warning for you, right? In this case, it's saying no. Suppose if it is going to tell, it may have some hepatitis related toxicity. So it's a warning for you. So if you are going to work on that molecule, so that it could be having some problems related to
I think today the participants need time to practice, and
Yeah.
tomorrow they will come up with more queries.
Yeah. So tomorrow morning, tomorrow first thing you can raise your doubts, or
Yes.
you can even, I think, list it out on the chat box. Then we
So
can
today's
go through it.
lecture, I think it was very much clear.
Yeah.
And sir explained beautifully that how to use the Swiss ADME software, and then we can go for these pharmacokinetic properties. So I
Yes.
think in today's lecture there was not any problem. Further we can practice. We are having tomorrow whole day, and we will come up with queries.
Yeah. And tomorrow I will talk about Pharma 4. So ideally you should have pi mole working, also the open babel. But don't practice when I am taking the class. Practice, because when I am taking and if you are practicing, then you will miss out what I am trying to say. But maybe we will have tomorrow half an hour gap. You can again practice. That way you will have doubts. You can ask me. Okay, not during my talk, but I will give a break so that you can practice. So that's called pharmacophore modeling. There you'll require pi mole and open babel.
So can we share the feedback link now in chat box?
Yeah, you can share with the.
Dr. Neha, can you please share the link of feedback link? Neha, are you here? Anyone, Neha, Chandan, Nisha, anyone of you?
Yes, ma'am.
Please share the feedback link in chat box.
Yes, ma'am. I'm sharing.
One request, participants, yes, Dr. Kavita, you will get all the recordings. And I also want to inform you that our college is having YouTube channel also. Whatever we conduct any event, that is automatically comes there. Anyway, we will record. We will give you all the recordings. We will share all the recordings. You please don't worry for these things.
Yeah, recordings we can share. Yeah, definitely.
Yeah. This feedback link is shared. I think 5 minutes or 5 to 10 minutes. You can take 5 to 10 minutes. Then we will close this form. Okay, thank you, sir.
Yeah.
Thank you, sir.
Thank you very much. Please
Okay.
practice, try some drugs
Yes.
which you know. That way you can use Swiss ADME, Zinc database, then the drug.com, ADMET, toxicity prediction. So all the databases you can use with some new drugs or new medicines which you know.
Yes, sir. Sir, see you tomorrow. Thank you
Yeah,
very
see you
much, sir.
tomorrow.